Welcome to The Explainer. Today, we're diving straight into a massive shift happening right now in cancer diagnostics. You know, it's pretty wild to think that some of our most powerful weapons against cancer aren't these giant room-sized hospital machines anymore. They're actually microscopic tools, literally smaller than a postage stamp. We're going to explore how these tiny molecular devices are just completely revolutionizing how we detect, understand, and ultimately treat cancer. It's an amazing journey from microscopic tech to massive, life-saving impact.
Picture this for a second. A patient is facing a brand new cancer diagnosis. It's an absolutely terrifying moment, right? And one of the very first questions that comes up is about treatment. But here's the really high-stakes dilemma. How do doctors know if a harsh, systemic treatment like chemotherapy is actually necessary for that specific person? We're finally moving away from the days of one-size-fits-all treatments, and we're stepping into a world of hyper-precise science that guides these major decisions.
And the real hero technology driving a lot of this precision, it's the DNA microarray. You might also hear them called DNA chips or gene chips. Essentially, these are tiny slides made of glass, plastic, or silicon. But there aren't just blank slides. They're printed with thousands of known DNA sequences. Just to give you a sense of scale here, scientists can pack tens of thousands of microscopic DNA spots onto a chip about the size of your fingernail. This tech literally gives researchers a window into the microscopic world of gene expression, letting them look at thousands of genes all at the exact same time.
So here's our roadmap for today's explainer. We're going to start with the cancer code, then look inside DNA microarrays. From there, we'll walk through the color-coded lab process, figure out making sense of the data, and finally wrap up with personalized medicine in action.
Let's start with Section 1, the cancer code and the high stakes of cancer care.
Okay, let's dive into this. The core problem we're facing in cancer diagnostics is figuring out the exact genetic changes that cause a patient's cells to divide uncontrollably. It's not just about finding a tumor anymore. It's about cracking the specific biological code driving that tumor. To do that, researchers need a way to compare what's happening inside a cancerous cell right alongside what's happening inside a normal, healthy cell. Specifically, they look at messenger RNA. Think of it kind of like this. If your DNA is the master instruction manual locked safely away in the cell's vault, messenger RNA, or mRNA, well, those are the active, temporary blueprints being sent out to actually build things. By comparing the active mRNA blueprints produced in cancerous cells against normal cells, scientists can spot which genes are overactive, which might be driving the cancer, and which ones are underactive, meaning they're failing to prevent it.
Section 2. Inside DNA microarrays, building the tool.
But how do you actually capture and measure thousands of invisible messenger RNA molecules all at once? That brings us to the anatomy of the DNA microarray itself. Long before any biological samples are even introduced, these powerful little chips have to be engineered. Specialized companies and scientists print them with highly precise, known sequences of DNA. We're talking really short pieces, usually just 20 to 70 bases long, spotted out in a perfect microscopic grid layout. Each one of those spots represents a specific, known gene. You can think of these tiny spots as highly specific molecular traps, just waiting there to catch their matching genetic material from the patient's sample. Section 3, the color-coded lab process, from cell to chip.
All right, let's step right into the lab to see how this actually plays out. This is the fascinating lab-to-life process, and we're going to break down the intricate 11-step sequence of microarray testing. It all starts with biological prep. Once they have the printed microarray and have collected both normal and cancerous cells from a patient, scientists isolate the messenger RNA from both sets of cells. Now, RNA is incredibly fragile. It degrades super fast. Because it's too fragile to work with directly, they use an enzyme to copy that RNA into a much sturdier, stable form called complementary DNA, or cDNA. Once they have that, they label these cDNAs with fluorescent dyes.
And this brilliantly illustrates how they keep track of everything using a really clever visual trick. The genetic material is extracted, copied, and then separated into two distinct visual categories. They dye the cDNA from the cancer cells with a glowing red fluorescent dye, and they hit the cDNA from the normal, healthy cells with a green fluorescent dye.
Next up is what's called the hybridization step. Those glowing red and green DNA strands are mixed together into a single solution, which is then poured right over the microarray chip. Because DNA naturally wants to pair up with its exact complement, those floating red and green strands will actively seek out and bind to their matching sequences on the printed chip.
Once they've had enough time to bond, we hit a really crucial step, washing. The microarray is washed to strip away any of those cDNA strands that didn't find a match. This ensures that only the properly bound, perfectly hybridized DNA actually stays attached to the glass slide.
Honestly, this washing step is absolutely vital to ensure accuracy in the final readout.
Let's just explicitly lock this in, because establishing exactly what these colors mean tells the entire story of the patient's cells. Remember this, the red fluorescent dye represents the genes that are active or expressed in the cancer cells. And the green fluorescent dye? That represents the genes expressed in the normal, healthy cells. Red is cancer, green is healthy.
To actually see the results, we move to the final physical lab steps. The microarray is scanned with specialized laser lights that cause those red and green fluorescent labels to literally emit their colored light. A computer captures these emissions and then merges the separate red and green images into one single comprehensive digital image that researchers can analyze.
Now, what's really interesting about this slide is how those colors combine when the computer merges the images. A pretty fascinating color story emerges. We don't just see red and green. If a spot is perfectly red, that gene is only active in the cancer cells. If it's green, it's only active in the healthy cells. But if both red and green bound to the exact same spot, they mix to create a yellow spot, meaning the gene is equally expressed in both. And if no cDNA is bound there at all, the spot just stays completely black.
Section 4. Making sense of the data. Balancing the scales.
So now we have this beautiful, multicolored grid, but we have to transition from the wet biology lab over to digital data crunching. Scientists can't just eyeball these colors. They must carefully and mathematically process this colorful data before a doctor can ever use it to make actual clinical decisions. The first major hurdle in this data analysis is called normalization. Simply put, normalization balances the scales. It compensates for the fact that the red and green dyes might naturally just have different fluorescence intensities. Without this crucial math, researchers could easily be misled if, for example, the red dye was just artificially brighter right out of the bottle than the green dye.
Let's move to the scatter plots and see how this builds mathematically. We use a technique called low-S normalization. If you look at the data before this is applied, the curve is heavily skewed and bent off the zero axis. It's artificially biased. But after applying low-S normalization, the data curve beautifully straightens out, so the centerline rests at a true balanced zero. Now, that data is trustworthy.
Section 5. Personalized medicine in action. The real-world impact.
With our data cleaned and verified, let's anchor this deeply technical analysis back to the human element. How does reading this microscopic gene expression actually dictate real choices for real patients? Let's look at a case study. Consider a patient recently diagnosed with stage 2 breast cancer. They've had surgery and are now weighing the terrifying choice of undergoing chemotherapy. Chemo carries severe, life-altering side effects. Fortunately, there's an FDA-approved personalized medicine tool called MammaPrint. It uses this exact microarray technology to analyze the gene expression of the patient's specific cancer cells to determine if chemotherapy is actually necessary for them.
The MammaPrint test takes all of that incredibly complex red, green, and yellow microarray data and translates it into a clear, actionable risk score. It categorizes the patient's biological profile as either high risk or low risk for metastasis. This gives families and doctors the power to make highly personalized, data-driven treatment decisions, and it often spares patients from grueling, unnecessary chemotherapy.
And cancer diagnostics are only getting more advanced. While microarrays are brilliant, researchers are already unlocking exciting, rapid advancements that will define what's next. Next-generation tools like surface plasmon resonance, or SPR sensors, are pushing the boundaries even further. These fiber optic sensors allow for the direct measurement of breast cancer markers straight from blood plasma. This totally eliminates the need for those complex cellular separation processes we just went through, making diagnostics way faster and highly specific for clinical settings. The versatility of these new plasmodic sensors is incredible. This technology won't just stop at traditional hospital labs. Because they're shrinking in size while increasing in sensitivity, they are soon going to power everything from localized lab-on-a-chip devices to routine environmental monitoring and even advanced telemedicine diagnostics directly in the home. We move from extracting microscopic bits of RNA to fundamentally altering how we treat major diseases. As biochips and genomic diagnostics become deeply integrated into our routine care, how will this microscopic data reshape the future of your own personal health decisions? It is a fascinating frontier. Thank you for joining me on this explainer, and keep learning.